So my presentation is about an injectable approach to the RaDVaC vaccine. So we all know that the main site of action of the RaDVaC vaccine is the nasal mucosal membranes, where it provides the mucosal protection against SARS-CoV-2 virus, mainly via IgA immune response. The aim of our study was to prepare an injectable form of the RaDVaC vaccine and to study its safety and efficacy, especially in terms of the systemic IgG and antibody response, as well as cellular immune response. We found that the injectable form of the RaDVaC vaccine is extremely safe and can be administered in both healthy people and people who had already been infected with COVID-19. The injectable form showed promising efficacy results. We continue to try to boost the efficacy of the injectable vaccine by testing different adjuvants, formulations, and epitopes. So first of all, how to prepare a sterile injectable formulation? So the injectable formulation must be prepared in a clean room environment under aseptic condition. In our case, compounding and filling was done in an sterile environment, class B, under laminar airflow, class A, to ensure sterility of the final product. The first injectable formulation was made based on Gen 8 and Gen 9 RaDVaC vaccines with a total of 22 peptide epitopes. The purity of all the peptide epitopes used were more than 98%, and all the excipients used were pharmaceutical grade. The formulation was similar to the nasal formulation with the following changes. Along with chitosan as adjuvant, we also used alum in the range of 0.2 to 0.5 milligram to boost the humoral immune response in the injectable formulation. Also, we used poloxamer P188 as a solubilizing agent to help solubility of the peptides. Also, benzyl alcohol was used as a preservative for the finished product. 0.2 micron sterile filters were used to filter all the ingredients before final mixing, and 0.45 micron filter was used for filtration of the compounded formulation. The following peptides were used for the injectable formulation. It includes all the peptides from Gen 8 and also some additional peptides from Generation 9 in RaDVaC white paper. For each vaccine, we use 5 microgram per dose of each peptide for the formulation. Mainly the spike proteins and also epitopes from nucleocapsid and ORF regions are there in the formulation. So to prepare the sterile formulation, we followed the same concept of the RaDVaC vaccine formulation. So we prepared the chitosan solution the same way that it is described in the white paper. Also the same for peptide solution. Here also we explain how to prepare the poloxamer P188 solution in the range of about 5 milligram per ml. And also how to dissolve the peptides in the poloxamer P188 solution. The preparation of TPP is exactly the same as what is described in a white paper. And for alum adjuvant solution, we also just simply dissolve it in the water for injection. All the water that was used for preparation was sterile water for injection, which is also, of course, pyrogen-free and endotoxin-free. For final vaccine solution, we used the laminar airflow biosafety class 2. We used sterile bottles in every activity. And all the ingredients were passed through the filters, 0.2 micron filters. And we mixed the chitosan working solution. We added the peptide solution. We added about 400 microliter of alum adjuvant. And also after that, we slowly added the TPP solution to form the nanoparticles. And at the end, we added benzyl alcohol. And then we adjusted the pH in the range of 3.5 to 4.0. And finally, we filtered the solution through 0.45 micron sterile PTFE syringe filters in a sterile container. I also would like to add that all the ingredients that we prepared before, we passed them through 0.2 micron filter to ensure the sterility of each ingredient. And then we filled the preferred syringes with a preferred syringe filling machine. We filled 0.5 ml of the vaccine solution in each preferred syringe in aseptic condition, and we ended with about 200 PFS syringe for the first trial that we made. The first study that we made on the vaccine solution was the particle size of chitosan peptide complex nanoparticle. So for that, we used a malware dynamic light scattering particle size analyzer, and the results showed that after this formulation, we had a quite uniform nanoparticle solution in the range of 150 to 300 nanometers of chitosan nanoparticles in the final vaccine solution. And the later studies showed that these nanoparticles are stable up to 1 month. Then for the safety of the injectable vaccine, we started to test it on the rabbits in a university lab, and we used both intradermal and subcutaneous injection, and followed the rabbits in the lab. Any sign of irritation, erythema, or edema up to 2 weeks after the injection, and we didn't see any of those things in the rabbits that were tested with the injectable vaccine. So it showed that in the rabbits and by administering intradermal and subcutaneous injection, there is no significant irritation, erythema, or edema. Due to the pandemic, and of course before many of the current vaccines were in the market, many colleagues volunteered to receive the vaccine. So we self-administered the vaccine. It was administered to the deltoid muscle as an intramuscular injection, and the injection was repeated after 3 weeks. More than 50 people participated in this trial. No serious side effects were observed in any of the volunteers who received the vaccine. In healthy people who haven't contracted COVID-19, the side effects were rare, and they include like temporary pain at the injection site, tiredness, and in rare cases, headache and muscle pain. In people who had already contracted COVID-19, the side effects were mostly the same, but more intense, and in some instances, it also included nausea as well. In general, the injectable vaccine was very much tolerable without serious side effects. Then we studied the efficacy of the injectable vaccine. The studies are still ongoing for both humoral and cellular immune response. For measuring humoral immune response, we used the S1-RBD IgG ELISA kit. The kit measures the level of IgG against the receptor binding domain of the S1 part of the spike protein of SARS-CoV-2. This antibody is also considered to be neutralizing and is a good indicator to measure the efficacy of the vaccines. Our studies showed that about 20% of the healthy volunteers who received the injectable vaccine and all of the recovered COVID-19 patients developed significant amount of antibodies after receiving the injectable RaDVaC vaccine. Of course, further studies now are on the way to boost immunogenicity of the injectable vaccine by increasing the immunogenicity of peptide epitope, such as IIT technology, and use of more effective adjuvants, such as squalene-based MF59, to induce proper humoral response in the vaccine recipients. So in the pictures, you see the samples of the people who had received the vaccine and some people who had also recovered from COVID-19. And the yellow is the intensity of the color, shows the degree of antibody. The more yellowish, the more antibody in the samples. For a study the efficacy of cellular immune response, we are performing animal studies, and these studies are now underway. We have acquired female 6 to 8 weeks bulb C mice for this study. 2 doses of the vaccine have been administered to the mice on days 1 and 21. 7 days after administration of the second dose, the spleen will be removed and will be analyzed by flow cytometer. Also, in some cases, 2 months and 4 months after that, the same procedure will be done for some other mice. For studying the cellular immune response, the following parameters will be analyzed by flow cytometer. The CD4 to CD8 ratio, CD8 immunophenotypes and functions, cytokine profiling, T cell proliferation in presence of spike SARS-CoV-2 antigen, evaluation of T follicular cells in B cell antibody secretion, evaluation of germinal center B cells. So after and upon successful completion of the animal studies, the human clinical trials will follow, and we will perform phase 1 of the human clinical trials. So as a conclusion, our studies show that the RaDVaC injectable vaccine is very safe to use with minimum and tolerable side effects. It does not have any biological contamination, hence it produces minimal allergic and autoimmune responses. Of course, further studies and trials are needed and underway to enhance the immunogenicity of the injectable vaccine, especially to increase the humoral immune response by using more effective adjuvants such as MF59 and modifications in the peptide epitopes such as IIT technology. The RaDVaC vaccine, injectable one, is already a very good candidate as a booster shot for vaccinated people as well as recovered patients. Our studies significantly show the increase of level of antibodies in the people who had already contracted COVID with administration of the injectable RaDVaC vaccine. Also, the injectable form of RaDVaC injectable vaccine can be easily modified to cover new and emerging variants by adding epitopes containing the mutations. And the RaDVaC injectable vaccine is also very scalable and can be produced in large quantities in many pharmaceutical plants around the world who can only produce any kind of injectable products. This vaccine can be produced there. So it's very promising. Further studies are needed. I would like to thank you all for listening to my presentation, and to contact me, please contact through info@radvac.org, and I'm also in the list of RaDVaC researchers. So please, through RaDVac, you can reach me, and I would be more than happy to ask your questions further in the conference. Thank you so much, and goodbye.
All right, well, good afternoon to you both, and thank you, Reza, for your presentation, and really for the work that you've done to really begin formal testing for various RaDVaC candidates, even in their adapted form, as an injectable. What we had originally envisioned, certainly was a mucosal vaccine, and there is still that potential, but it shows the leverageability and the adaptability of the platform, that it can be adapted to, these alternative models and the alternative routes of administration. So I think the two of you probably have questions to answer. So I'm gonna close my video out and allow you to a few minutes just to speak with each other.
So thank you
Okay,
very
thank you
much,
so much,
Dr.
Alex.
Mostofi. That was excellent. I wanted the RaDVaC community to see your presentation because you are an ideal representative of the effort that we hoped people would make after we made the initial investment of designing vaccines and releasing from the world. You've done exactly what we had idealized. You've modified it. You've used it to your circumstances. You've used an injectable vaccine because you thought it was more appropriate. You're doing a placebo-controlled randomized clinical trial, even though we favor challenge trials. This is part of the big tent approach that we think is really necessary to create a very different kind of open science model relative to the patent-dominated pharmaceutical model that has got us in the situation that we're in now. So anyway, I just wanted to thank you very much, not just for joining the RaDVaC network and doing the great work that you've done, but in creating the variations on what we've done and carrying the work forward.
Thank you so much, Person. It was really a very good opportunity for us to explore this new open source way of manufacturing vaccines. Especially we started this when vaccines were not available. You know that there is still a big shortage of the vaccine in the market, and it will continue for a long time. We are seeing the emerging variants every now and then, and we should be prepared for that. Other thing is that the booster shots, because many people, especially in the United States, they have received their first shot, but sooner or later they need the booster shot, especially when we consider that the new variants are coming. So this problem will be with us for a long time. And I think that the RaDVaC vaccine is a very good solution, is easily adaptable for the new variants, and can be manufactured very fast, and also is highly scalable. So as I said, we think that there is much effort that is needed for boosting the immunogenicity of the vaccine, but we already have also proved that for the people who have already received the first dose of the vaccine, or they have been infected with COVID, the RaDVaC vaccine will significantly increase the level of antibodies. And with antibodies, we mean the neutralizing antibodies, as you have chosen the peptide epitopes that are for the RBD part of the spike protein. And even we know that also you adopted the variants, for example, the UK variant, you also adopted those on the design of the epitopes. I think that the future is very good for this vaccine. It can be produced in larger scale and very fast, and we really look forward to more study on this. And this is why we actually are doing a complete trial, animal studies, both in humoral and cellular immune response, and with whatever that is needed to register this vaccine, even in the WHO platform. So we are going towards that goal, and I think that very soon we are going to register this as a WHO vaccine as well. So we really look forward for all the contribution from all the partners in the RaDVaC researchers group, and all the inputs, and also of course your expertise. So I think that the future is very good for this vaccine, and it can be a very good candidate, especially when we are also talking about like this pan-coronavirus vaccine. It can be also adopted for that purpose as well. Having said that, I think we are very confident to continue this way. We will share the results as we move forward, and So we would like to have the contribution from all the partners in the RaDVaC project.
Well, you're certainly a model contributor, and I encourage all people out there who whose countries and regions of the world who need vaccine and who need a better plan for the future to use Actoverco's and Reza's model for the adoption of the RaDVaC vaccine as a way to break away from the constraints of patents and and typical strangleholds on the high concentration of vaccine power in a very small number of organizations and a very small number of countries. So I just want to mention very briefly, because I know people probably were wondering, seeing that there's a big difference between the immune response in the previously uninfected and the infected, where you get only 20% of people responding to the vaccine versus people who had previous infection. Everyone is getting a very high titer. I think a big part of that difference is that Actoverco, like all pharmaceutical companies, needs a minimal dose regimen to keep compliance high, right? So you've used a 2-dose regimen of a prime and a single boost, whereas we know that chitosan, and we've known this from the very beginning from the clinical trial data that we looked at, is a mild to moderate immunostimulant. So we've actually recommended minimum of 3 booster doses for every one of the generations of vaccine that we've released. So that is one of the reasons, reducing the number of boosters is one of the reasons that you and others need to investigate the use of more potent adjuvants, is so that you can keep the number of doses low. But don't, for people who are watching this and who will be watching this recording, don't be afraid of the low effectiveness or low immunogenicity of the unmodified chitosan vaccine. It is, I'd actually think it's a good sign to show that the vaccine, even without additional very strong adjuvants, is working, even in previously uninfected people. But there's a lot of room for improvement, which in PanCOV and Gen 10, we've already achieved